In the last couple of segments, we looked at general features of enzymes, essential catalysts of chemical reactions that enable life. In this video, we are going to dig a bit deeper into enzymes by looking at some of the main categories of enzymes. Although there are literally millions of different enzymes, these can be grouped based on the general type of chemical reaction that are catalyzed. In this video, we focus on how we look at a particular chemical reaction and can match that chemical reaction to the general type or class of enzyme that would catalyze that reaction. Let's go ahead and take a closer look. When we are classifying enzymes, in general, we're going to classify them based on the type of reaction that they catalyze, where if we think back to organic chemistry and the general types of reactions that we learned, we can generally bucket reactions into several different categories, such as oxidation and reduction reactions, or hydrolysis reactions, where we used water to break a molecule. We looked at isomerization reactions, where one isomer becomes another, and a variety of other examples of these types. So in general, we're going to classify our enzymes based on a few different types of chemical reactions. Specifically, there are 6 classes of enzymes that we will discuss here. Those 6 classes of enzymes are oxidoreductases, which are going to catalyze oxidation or reduction, as the name oxidoreductase implies. Transferases, which will catalyze the transfer of a functional group from one molecule to another, hence earning them the designation transferase. Hydrolase, with that hydro for water embedded, are reactions where the enzyme catalyzes the use of water as a reactant to break a molecule into 2 separate final products. Lyases, another classification of enzyme, are enzymes that catalyze the adding or removal of a group without using water as a reactant. So while hydrolases used water as a reactant to result in the removal of a group, lyases enable that breakage of a bond without using water as a reactant. Isomerases are another important classification category of enzymes. Those are enzymes that catalyze rearranging atoms in a molecule to yield an isomer, meaning rearranging them to yield a stereoisomer or rearranging them to yield a constitutional isomer. Ligases are enzymes, finally, that catalyze the formation of a bond between molecules by using a molecule called ATP, adenosine triphosphate, as a source of energy to help drive the reaction. So in this video, we will dig into more detail and look at examples of each of these 6 classes of enzymes. One key thing you will notice as you look at all of these enzymes is that all of these classes of enzymes end in the suffix ase, and that is very common amongst enzymes that when specifying classifications of enzymes or even a specific enzyme name, ase is a very common suffix that allows us to quickly recognize a molecule as an enzyme, as a protein that catalyzes a chemical reaction. Now let's describe in more detail each of these general classes of enzymes, including examples of how to recognize each of these in action. Oxidoreductases are enzymes that catalyze oxidation or reduction. Remember from earlier discussions that oxidation refers to an increase in the number of bonds between carbon and an electronegative atom, such as oxygen. For example, if we go from an alcohol to an aldehyde or ketone with a carbonyl group, we have increased the number of carbon-oxygen bonds from 1 carbon-oxygen bond to 2. Therefore, we describe that as oxidation. Reduction is the opposite process, where we are decreasing the number of bonds between carbon and an electronegative atom. So in other words, if we went from an aldehyde or a ketone to an alcohol product, we would describe that process as reduction. In the case of oxidoreductases, one key example of this class of enzyme in action occurs in the metabolism of ethanol and other alcohols. If we have ethanol, CH3CH2OH, shown here, and that is presented with the correct oxidoreductase that recognizes ethanol as a substrate, and there are oxidoreductase enzymes in the liver, in particular, that are able to bind to an ethanol substrate, and they act to oxidize that ethanol substrate during metabolism of alcohol to first an aldehyde. This molecule is called acetaldehyde because it has 2 carbons. And then oxidoreductase further comes in and catalyzes the further oxidation of the aldehyde to form a carboxylic acid product, specifically acetic acid, which is highly polar and can be passed out of the body through the urine. Transferases are the next group of enzymes that we'll focus on. Transferases are enzymes that catalyze the transfer of a functional group from one molecule to another. One example of this is the illustration of how alanine can be transformed into glutamic acid. Let's take a look at how this reaction of alanine plus a compound called alpha-ketoglutarate work via catalysis by a transferase enzyme. Alanine, one of the 20 standard amino acids, has, of course, the carboxy group, COO-. I'll show this as the Zwitter ion form. And then we have our alpha position, where our carbon here is bonded to our R group, which in the case of alanine, the R group is a methyl group, CH3, and then finally connected to that yellow alpha CH group is our N terminal, our amino group, which I'll show as N+. So this is our structure here of alanine. Alanine reacts with a molecule called alpha-ketoglutarate. Let me go ahead and draw that out. I will draw that as the Zwitter ion as well at physiological pH. So we have at the left-hand end of this molecule COO-, followed by a carbonyl group. After that carbonyl group is a series of 2 CH2 groups. So I'll show CH2 in parentheses and then put subscript 2 to indicate there's 2 of those CH2 groups, followed by another carboxylate group, COO-. This, which I have drawn here, is the structure of alpha-ketoglutarate. When these two are bound to a transferase enzyme that has substrate specificity for each of these 2 substrates, what happens is that the transferase will act to catalyze the transfer of the amino group, circled here, from alanine over onto the carbon where the carbonyl was of the alpha-ketoglutarate. The resulting structure from that reaction ends up being glutamic acid. And I've drawn here the glutamic acid using exactly the same color code that I did on the reactant side of the equation, where the NH3 group of alanine is transferred via transferase to where the carbonyl group was of alpha-ketoglutarate. So everything in pink here derived from alpha-ketoglutarate in the skeleton of the glutamic acid. The group in yellow derived from alanine. So in other words, we transferred a group from alanine onto alpha-ketoglutarate to yield glutamic acid. Now you might ask, well, what is the other product of this reaction? We can draw out the other product of the reaction with the leftover atoms here. That is, from alanine, we have not yet used the CHCOO- and that green R group, our methyl group here. And so if we draw that out, that is going to become a portion of our other product of this reaction. So we have CCOO-. We used this H from CH to balance the equation over here in yellow and glutamic acid. So that C from alanine is still bonded to the CO-, it's still bonded to the methyl group, which I'll put in green, CH3, and then the last remaining piece to fill the octet is that carbonyl from alpha-ketoglutarate right here becomes a carbonyl group on that, what was originally the alpha carbon of alanine, becomes a carbon that's bonded to the carbonyl donated from alpha-ketoglutarate. So that carbonyl group, in other words, is transferred from alpha-ketoglutarate to alanine during this process as well, to give this as the side product of the reaction that transforms alanine by reacting with alpha-ketoglutarate into glutamic acid. And this is a way that the body is able to generate more glutamic acid in the event that there's plenty of alanine but not enough glutamic acid to build the proteins that we need. The next stop on our tour of enzyme classification is the class of enzymes known as hydrolases. From the term hydro, we imply that water is involved in these chemical reactions. And indeed, hydrolases are enzymes that use water as a reactant to enable the breakage of covalent bond or bonds within molecules to break them into multiple products. One example of a situation where a hydrolase is involved is in the breakage of peptide bonds. Here I have shown a simple dipeptide with one of the two amino acid units shown in pink and the other shown in yellow. This could be a peptide of any length here. In these cases, what happens is we previously talked about a group of enzymes called proteases, which break these peptide bonds. Proteases are a type of hydrolase because they use water to react with a molecule, resulting in breakage of this covalent amide linkage here to release, on the product side, the cut amino acid units shown in pink and shown in yellow to match the colors of the units on the reactant side. Both the peptide and water bind at the active site of the enzyme. This allows the peptide and water to come into very close proximity to one another, and the enzyme active site also allows the water to line up perfectly with the location of this amide linkage for the necessary bonds to be broken and formed. Effectively, this will also lower the activation energy required for the reaction, therefore favoring the breakage of specific peptide bonds within peptides and proteins to enable ultimately the release of the free amino acids through this type of enzyme that's broadly classified as a hydrolase due to the fact of it using water to break covalent bonds. Now we move on to the next classification of enzymes, which are lyases. Lyases catalyze the addition or removal of a functional group from a molecule without using water as a reactant. So a key distinction here between a hydrolase and a lyase is just that the lyase works without using water as a reactant. So if you see water reacting to break a chemical bond, we classify it as a hydrolase. If you don't see water present and you see a molecule being broken into two, then we describe it as a lyase. One example of a lyase is in the reaction of pyruvate, and pyruvate is a key intermediate in carbohydrate metabolism that participates in the citric acid cycle and others that we will look at in the next unit. And what happens with pyruvate is there is an enzyme that is classified as a lyase that reacts pyruvate with an acid that also binds to the active site of the enzyme to create acetaldehyde by breaking the bond to the carboxylate group shown here. That releases acetaldehyde by placing a proton in place of where the COO- broke away from the molecule. The other product of the reaction, to balance it out, is carbon dioxide, which floats away as a gas from the system and is one reason why CO2 is a byproduct of metabolism that we exhale. That particular reaction, without the action of a lyase-type catalyst, would be very slow, and the body would not be able to control it, would not be able to undergo respiration at the rate necessary for us to sustain life as we know it. Our fifth stop on our journey through the different types of enzymes is with the isomerases. Breaking this word down, ASE, as we've seen, refers to an enzyme. Isomer, you may recall from earlier units, refers to two structures that have the same number and type of atoms, but a structural difference, where that structural difference could be that the group has switched locations entirely within the molecule from one location to another, giving a completely different name, perhaps even a different functional group. In that case, isomers are referred to as constitutional isomers. Isomers can also be so-called stereoisomers, which are ones that have the atoms in the same order within the molecule, but they have a different three-dimensional shape, such as an R stereoisomer becoming an S stereoisomer. One example of an important isomerase is the one that catalyzes the interconversion of the stereochemistry of amino acids, particularly in bacteria. Although we recognize that the 20 standard amino acids usually have an L configuration, they can have, in some cases, a D configuration catalyzed by an enzyme that is prevalent in some types of bacteria. In particular, bacteria that produce a peptidoglycan cell wall, in other words, gram-positive bacteria, will frequently have enzymes that convert L-alanine and L-glutamate into their D stereoisomers, in other words, D-alanine and D-glutamate. These D configuration amino acids are what enable cross-linking of different structures within the peptidoglycan cell wall to give the cell wall its rigid structure. Our sixth and final stop on our tour of enzymes is the group of enzymes known as ligases. To ligate means to link together. So what ligases are going to do is catalyze bond formation or bond linkage using ATP, adenosine triphosphate, as an energy source. ATP is a biomolecule that is widely used in cells to take reactions that would otherwise be endothermic and allow them to be exothermic because ATP has a high energy phosphate bond that can readily be broken and releases lots and lots of energy. In other words, the breakage of ATP is a very exothermic reaction that can be used to help drive other less energetically favorable reactions forward. So in the case of these types of reactions, what happens is that ATP binds at a site on the enzyme. Pyruvate, in the case of our example here, and CO2 bind at the active site, and that results in the formation of oxaloacetate, shown here, by linking the CO2 at this right-hand end of the pyruvate molecule. In other words, we're catalyzing bond formation here by linking CO2 to the pyruvate molecule. That requires ATP as an energy source because this reaction of converting pyruvate to oxaloacetate would otherwise be an energetically unfavorable reaction. When we break ATP apart, I show as a product of the reaction adenosine diphosphate because the breakage of one of the three phosphate groups of triphosphate is what results in the release of energy to help drive this reaction of converting pyruvate into oxaloacetate. This is an important reaction in metabolism because, as we saw earlier, pyruvate is one of the intermediates in metabolism. So with that, we conclude our journey through the 6 different classifications of enzymes. Now you should be able to take a variety of different types of chemical reactions that are catalyzed by enzymes and classify them into one of these 6 categories.